As the final speaker of this symposium, we are pleased to welcome a special guest speaker from abroad, Dr. Viorica Bondici from the Canarian Light Source. So Dr. Viorica Bondici is a beamline scientist at the Bio-Imaging Hard X-ray Microprobe beamline at the Canadian Light Source Synchrotron Facility. She leads the beamline operation and development initiatives. She got her PhD in applied environmental microbiology from the University of Saskatchewan and used synchrotron techniques to understand biogeochemical processes. So her expertise includes a multi-resolution synchrotron-based XRF imaging, X-ray spectroscopy, and experimental design for complex samples, and she actively collaborates with diverse user communities to integrate the complementary synchrotron techniques. So the title of her talk is Integrating Multi-Resolution XRF Imaging for Elemental Analysis in Agricultural Science. Please.
Thank you for the lovely introduction. So this is the short overview of my presentation. So why synchrotron techniques matters, what type of insight they can offer in agricultural science. And also I will touch upon the principle of X-ray fluorescence imaging for potentially new users, and what factors to take into consideration when it comes to select a beamline of interest. And in addition, describing the capability of the bio-XCS imaging beamline, I will also cover some technical aspects of the beamline that can be taken into consideration regarding sample feasibility or data collection planning in agricultural science. So the Canadian Light Source is the only synchrotron facility in Canada. It is a third generation facility. It houses 22 synchrotron beamlines, and the beamline's energy ranges falls within a very wide ranges, including infrared, soft X-ray, tender, and hard X-ray region. So synchrotron-based visualization can provide a very powerful and non-destructive insight, for example, into plant health or crop yield, or it can be used to study or extend shelf life of crops or seed processing, and also can offer insight into disease resistance and sustainability. And in particular, X-ray fluorescence imaging can provide information, for example, infectious routes, or understand the micronutrients dynamics in plant tissue, enabling us as researchers for targeted breeding or to select the traits of interest. Or also we can improve, such as treatment for diseases, or increase the efficiency of nutrients uptake that serves sustainability. So just very briefly regarding the principle of X-ray fluorescence imaging. So when X-rays interact with matter, one of the events is photoabsorption. And then as a result, an electron from the inner shell is ejected into continuum. And then this configuration of the atom is very unstable. And as a result, an electron from the outer shell would immediately fill the empty core hole, prompting an X-ray fluorescence photons emission. And this is an experimental plot to indicate the importance of the energy of the incident beam. So if the energy is below an absorption edge of an element, you would just measure scatter peaks. However, in this case, sorry about that. In this case, if it's above the absorption edge of an element, such as selenium K edge, then you would also measure the specific emission peaks for selenium K alpha and K beta as well. So how these images, X-ray fluorescence images, are built. So the synchrotron beamline components would deliver a monochromatic small beam onto the sample, and the sample would be rastered in front of the incident beam. And the emitted X-ray fluorescence photons at each pixel or steps would be collected or measured by an X-ray detector. And I would like to note that, that the energy of the emitted photons are unique for each element, therefore would serve as a fingerprint to identify and actually study the distribution of elements non-destructively in very wide type of samples. So the images, the XRF images are built from a single pixel X-ray fluorescence spectra, where the peak would represent the energy of the emitted photons of the elements present of the sample, and the height of the peak would represent the number of photons at these energies. And then from the sum of spectra, each element can be deconvoluted, and you generate the elemental distribution images. So where to start, how to select a beamline? These are just a few factors that I'm mentioning, and these are not limited to these examples, but first would be at what energy ranges your element absorption energy would fall into. It is in soft X-ray, tender or hard X-ray region. What type of techniques you would actually resolve your research question? Is it imaging, spectroscopy, diffraction or tomography? And what type of resolution you need? You need the bulk spectroscopy or macro, micro, or nano resolutions. I would like to present this project to actually indicate the importance and benefit, and basically the inherent strength of multi-scale X-ray fluorescence imaging. So in this study, researchers were interested in the bioaccumulation of mercury in a model organism zebrafish that was subjected to acute concentration of methyl mercury. And then they utilized a macro mode, a 20 micron resolution, to image the whole organism specimen. And then they used the micro resolution of 5 micron and submicron resolution to image cross sections of relevant areas. So the macro resolution helped to identify the location where mercury was preferentially accumulated, identified the eye as a sensory organ, and then the micro mode on the relevant cross section of the head cross section revealed that mercury was preferentially taken up by the epithelial cells surrounding the eye core lens. And then the submicron resolution further revealed that mercury was taken up by the photoreceptors within the retina. So the project, this multi-scale approach, revealed and identified an important questions where methyl mercury is accumulated, so identified the eye sensory organ, and it was also further proposed that visual disruption due to exposure to high concentration of methyl mercury could arise from the direct damage of the photoreceptors in the retina. So at that time, this project was carried out at multiple beamlines and 2 synchrotron facilities, and really embodied the vision to have a multi-scale resolution XRF imaging capability beamline. So the BioXCS imaging beamline stands out in the synchrotron communities, having such a capability, but being part of a BioXCS sector, users would have access to complementary techniques, high sensitivity spectroscopy and bulk spectroscopy techniques as well. So these 3 beamlines within the BioXCS sector shares the primary optic enclosure. However, they have their individual beamline components, and they can operate independently from each other. So just looking a little bit closer of the main beamline components. So the BioXCS imaging beamline has an in-vacuum undulator. We have a collimator mirror, and then a fixed exit mono with silicon 111 and silicon 220 crystals, and then a focusing toroidal mirror, and the KB mirrors are utilized downstream to focus the secondary source. So the energy range of operation is 5,000 and 20,000 EV, and the current mode of operation is macro mode with beam size available between 20 to 100 micron, with flux reaching 10 to the 12 at the bigger beam sizes. And in micro mode, we generally operate at 5 micron resolution with a flux of 10 to the 11th order of magnitude. We collect data, imaging data in bidirectional fly scanning mode, meaning as the sample is moving in both directions, the data is being collected, and we have dedicated detectors for both setups. So looking closer in the hatch, how it positions, so macro mode is positioned close to the secondary source, and then we are utilizing KB mirrors to focus the secondary source downstream. The main techniques of the beamline is X-ray fluorescence imaging. Basically, we are rastering an area with a small beam and obtaining X-ray fluorescence imaging data. And then this technique can be combined with micro XAS that actually provide information, speciation information of the element. And basically, you would be targeting a point, and instead of rastering the sample, you would raster the energy across the absorption edge of the element. Zane's imaging is also available. Basically, these are multiple XRF images of the same area. However, these are across incremental energy points and would provide you cluster of images of different spectral features. So I would like to show a little bit more in detail of the macro mode setup. We are utilizing pinholes to aperture the beam to different sizes. These are basically laser drilled pinholes in heavier atom materials that are not generally present in biological sample. And I would like to highlight that we can operate in 2 configurations depending on the thickness of the sample. So if the sample is several millimeters thick, we can operate at 90-degree stage configuration, and the detector is at 45 to reduce the parallax or the shadowing effect. And then we can operate at 45-degree stage configuration where the detector is at 90 degrees to dampen or reduce the scatters, and the samples would be in several microns or a few microns thickness. So since we have a pretty large sample holder, we can accommodate different sizes of samples as large as 21 by 21 centimeters. So this is an example where researchers were interested in the location of selenium and its speciation in our pollinator, the bumblebees. They imaged the bumblebee at 90 degrees stage configuration and at 40 micron resolution. And they also were interested how the microbiome naturally present in the bumblebee gut could play a role in the detoxification method by changing the redox states of selenium. So large areas can be readily scanned by changing the beam spot sizes, just changing the pinhole sizes, and more details analysis can be obtained by moving from a larger beam size to a smaller beam size without going into the hatch and changing physically the setup. These pinholes can be changed remotely as desired. So this is the micro mode setup. We are utilizing the KB mirrors to focus the beam vertically and horizontally. And we also have both stage configuration available. And the sample frame size could be as big as 2 centimeters vertically and 3 centimeters horizontally. So this is an example of a project that utilized the micromode setup. The researchers were interested in micronutrients distribution in oat seed cross-section. And then the study revealed that zinc and manganese, the micronutrients, were distributed close to the alluvium layer of the seed, providing very important information regarding seed processing. So to avoid harsh hulling, to prevent the removal of the nutrients. And this is another example where how we utilize XRF imaging for comparing specific areas and also utilizing it to quantify elements at specific areas. So the researchers were interested in elemental distribution in the embryo and the alluvium region in rice seed cross-sections in the germinated and mature seeds. So we have a level of automation when it comes to data collection. So we can mount several samples in macro mode as well as in micro mode. And through coarse scanning, basically, we can get an overview of all the sample present, and it can guide us the selection and queuing up multiple fine scans regions. And there would require only minimal user input when it comes during the data collection process. That could be several hours, overnight, or several shifts. So I would like to present a couple of projects that utilize the multi-scale XRF imaging approach for their studies. This particular study was interested in nitrogen uptake efficiency in the soybean root nodule. So they scanned the whole root system in macro mode of 90-degree stage configuration at 40 micron resolution. But as you would imagine, these nodules are sensitive to desiccation, so the scan needed to be carried out pretty fast at 20 millisecond dwell time. And they had a closer view of the elemental structural information of the nodules at 20 micron resolution at the same setup. And also they imaged the cross sections of these nodules at 5 micron, and through oversampling, they obtained a 20 micron resolution of the cross sections. And they concluded that iron was mostly distributed in the central infected zone, and zinc was distributed in the vascular system. So this is another example that used the multi-scale XRF imaging approach in combination with micro-XAS to obtain speciation information. So they scanned a relatively large area of the wheat that was grown in selenium-enriched soil. And this macro-resolution image guided them to select a sub-region for final resolution images. And then they selected specific points to obtain speciation information of the selenium. So we receive projects from all the research pillars, including health science, such as biomedical imaging. In general, we receive users are interested to scan a cross-section of animal models that are to study such as neurodegenerative diseases such as Alzheimer's or multiple sclerosis. And also in environmental studies, this example is to study contaminants found in the environment, such as arsenic, how they co-occur with other elements and their speciation that would provide information regarding their solubility or their state in the environment. And we receive a significant number of projects from agricultural science. So the bioXCS sector really works complementary with other beamlines at the Canadian light source facility. And I would like to showcase a study carried out at cross beamlines to analyze the structure and cellular details and elemental distribution in canola seed. And as you can see, the tomography beamline provided structure information at coarser and higher resolution, and XRF imaging provided a bioXCS imaging beamline elemental distribution that complements very strongly with the mid-infrared spectroscopy beamline data, where you obtain information about the macromolecule distribution, such as protein, carbohydrates, and the lipids. And higher resolutions at the soft X-ray region, a 20 nanometer resolution, this yielded the visualization of the phosphorus clusters at this magnification level. And I would like to also highlight that the users have access to the scanning electron microscope technique, imaging technique that strongly complements with the other beamline techniques. We have very well-established groups that support science. The strategic support group, they played an important role to establishing the agriculture user community. And then also the industry service group provide services from all the scientific fields, and also including the agriculture and research. And then I would like to acknowledge Drs. Graham George and Ingrid Pickering, who is now in the audience. They were the recipient of the funding to build the bioXCS sector. And I would like to acknowledge all the engineers and developers and the senior scientists who played role in building and commissioning this beamline. And I would like to thank you for your attention.